First of all, I would like to thank Dr. Aleksandar Kostic for his participation in this conference and for giving us the opportunity to share with us his knowledge and his experience on the interaction of the microbiota with the human body and health and diseases. I'm sure that this conference will give us great teaching and a lot of reflections. Thank you very much, Alexander, for this great honor of listening to you and for being a great collaborator. I'm going to allow myself to read your bio sketch in Spanish. El profesor Alexander David Kostic es uno de los grandes investigadores jóvenes. Obtuvo la licenciatura en Biología Molecular en la Universidad de Toronto y el doctorado en Ciencias Biomédicas y Biológicas en la Universidad de Harvard. Realizó un postdoctorado en el Broad Institute del MIT y Harvard, trabajando en el campo de microbiología, el metabolismo y la biología computacional. Su principal interés de investigación es el descubrimiento y caracterización del mutualismo humano-microbiota desde una perspectiva evolutiva, metabólica e inmunológica, con un enfoque particular en cómo estas relaciones pueden ayudarnos a comprender mejor las enfermedades metabólicas y autoinmunes. Sus contribuciones científicas desde sus inicios incluyen descubrimientos de asociaciones novedosas entre el microbioma y la enfermedad humana, que han llevado a una nueva comprensión de las interacciones microbioma-hospedero en el cáncer colorectal, la diabetes y aún en el ejercicio. En su laboratorio se adopta un enfoque multifacético para estudiar el microbioma mediante modelos de diabetes en ratones notobióticos, el desarrollo de algoritmos para análisis multiómicos, el cultivo de diversas bacterias simbióticas humanas y la ingeniería de estos microbios a través de biología sintética. Ha recibido varios premios y reconocimientos. Él es el creador y director del curso de literatura sobre microbiomas, así como instructor de varios otros cursos que atienden a estudiantes graduados en inmunología, biología computacional, redacción de financiamientos y ética de la investigación. Es cofundador y miembro de los consejos de administración del Deep Biome Therapeutics Incorporation y del consejo asesor científico de Fit Biomics. Su objetivo final de investigación es desarrollar nuevos métodos para diagnosticar y tratar la diabetes mediante la comprensión de las diversas capacidades funcionales de la microbiota simbiótica y su impacto en nuestro metabolismo y sistema inmunológico. Desde 2016, él es investigador asistente en el Gelding Diabetes Center y desde 2017 es profesor asistente de microbiología en la Escuela de Medicina de Harvard. Ha publicado varios artículos de investigación en revistas como Nature Medicine, Cell Host, Microbe. Según Google Scholar, tiene más de 13,500 citas. Muchas gracias, Alexander. Thank you very much, Dr. Kostic.
Hi, everyone. I'm very excited to be here today. I'd like to thank Lorena for her kind invitation. I've visited her and her group at Imagen before in Mexico City and had such an amazing time, so many great memories with such fantastic hosts. I hope to return again some point in the future, but until then, I'm happy we're still in touch and that I'm able to present at your seminar today. Thank you again for the invitation. And I'm going to tell you about work in my lab, which is focused around the microbiome and its role generally in diabetes, both type 1 autoimmunity and type 2 metabolic diabetes. And trying to understand from the microbiome why the incidence of diabetes has been increasing so drastically in recent years. So this is a graph of the incidence of infectious diseases over the past several decades. And as you can see, nearly all infectious diseases are decreasing. Of course, we're in the middle of a pandemic, and so this isn't a problem that has disappeared, obviously. But generally, we can say very confidently that over many decades, we've really built a grasp around infectious diseases through antibiotics, the development of antiviral drugs, through sterile practices in the hospital, through hygiene in general, and sanitation, and a lot of cleanliness in the home and at work and around our communities, etc. All of this has resulted in a much lower incidence of infectious diseases. But now, if we look at the incidences of immune diseases over time, and this is including autoimmune diseases like type 1 diabetes and also allergic diseases like asthma, the incidence of these have been steeply increasing over the same span of time, over these past several decades. And is it possible that there's some connection here? So this is what it looks like from a temporal point of view. If we look at this now in terms of geography around the world, and we ask, where do serious childhood diarrheal diseases happen? They happen in Africa and Asia, and in general in lower income countries relative to the United States and Western Europe. Now, if we look at where the incidence of type 1 diabetes occurs, the map is almost the mirror opposite. The incidence of these diseases are so low in areas where childhood diarrheal diseases are high. And again, I ask, is there a possible connection here? And what could it be? And what I propose this is really the hygiene hypothesis, which states that early hygienic practices lead to incidences of chronic diseases later in life because of the lack of exposure to microorganisms in early life as the immune system is developing. And that these early exposures to diverse organisms are critical to the proper development of the human immune system. And so obviously, when we kind of think about this today, we think about it in the context of the human microbiome that inhabits our gut and our skin and other parts of our body that, of course, they're not pathogens. They are symbiotic bacteria, commensals, and even mutualists that we believe play an important role in keeping us healthy. And to understand the importance of the microbiome, we need to only look at what life is like in the absence of a microbiome, which we can observe in our germ-free animals, grown in a completely sterile environment, no detectable fungi or bacteria or viruses. And although they do survive and can live, they have a large plethora of health conditions. They have to consume more than 2 times as much calories as a conventional mouse just to maintain its body weight. Very prone to autoimmune and allergic diseases, which is why they're a great model for type 1 diabetes, for example. And they have many neurological and behavioral issues that are quite striking. And so what's even more fascinating is that when you colonize these mice at an early age with a normal mouse microbiome, now you can rescue all of these phenotypic defects. And so it just kind of underscores the importance of the microbiome. So that's in animals, of course, but is this important in humans as well? And so to get at that question, understanding the role of the microbiome in human immune development, we conducted a multi-country study. This is the dive immune cohort, which was based out of Finland, led by Mikael Nip at the University of Helsinki. And it is a multi-country cohort of children that are at very different risks for developing type 1 diabetes. The incidence of type 1 diabetes is higher in Finland than anywhere in the world. And if you look just across the border to Russian Karelia, the incidence is 8-fold lower, despite the fact that they are geographically very close to each other. They have many cultural similarities. This is a border that's moved back and forth over the wars, and very similar genetics. And so what accounts for these differences? Again, this ties into the hygiene hypothesis, and the big difference is the GDP of these countries, especially in this region of Russia, around Petrozavodsk, and the Karelia region. It's a very poor region, and Finland has one of the strongest economies in the world. And this means that the lifestyle of the people is very different. People in Finland live a lifestyle in urban environments with sanitation in the homes, very cleanly environments. People in Russian Karelia are much more often live in rural communities, have exposure to animals, farm animals, and soil, and generally have less sanitation than in Finland. So that means that probably the microbial exposures are much higher, particularly at an earlier age. So we also included in this cohort Estonia as kind of a control. It is former Soviet bloc country, and as its GDP has risen, its incidence of type 1 diabetes and other autoimmune diseases is also rising. So this is just a principal coordinate analysis on all of the samples. These are gut microbiome samples, and I didn't show in detail, but this is actually a cohort of 75 infants from each country, 75 from Finland, 75 from Estonia, 75 from Russia, and we're tracking their microbiome from the time they're born until they're 3 years old. And this is all of the gut microbiome samples that we collect approximately monthly. And what you see is that there's not a big difference between Finland and Estonia, but the Russians in red look quite strikingly different. They're kind of focused towards the bottom of this graph here, suggesting a big difference in the overall community composition in their microbiome. To look at it in a little bit more detail, here I'm showing the full phylum level composition of the microbiome. And what you'll notice is that after 12 months, the composition is quite similar across these countries, not very different, but in this critical first year of life, you can see these very pronounced differences in the overall composition. The Russians are dominated by bifidobacteria, which belong to the phylum Actinobacteria. Also have a lot of Firmicutes, but the Finlands and Estonians are dominated by Bacteroides, which is much less common in Russia. And so now let me propose one of the reasons why I think this difference exists, and it all comes down to human milk oligosaccharides that come from mother's milk and are utilized by the infant's microbiome. These oligosaccharides actually cannot be directly metabolized by the infant itself. And it's believed that the only purpose of these oligosaccharides is to cultivate a microbiome in the infant. Certain bacteria are kind of have this job of being the HMO utilizers, and generally they're Bifidobacterium and Bacteroides. So then what we did was measure the HMO gene content across these cohorts and ask which microbes did they come from. And for the most part in Russia, this role is played by Bifidobacterium, whereas in Estonia and especially Finland, this role is almost completely performed by Bacteroides, which again is consistent with the phylum level differences we see. And so as to why Bifidobacterium plays this role in Russia, I don't have a good answer. It might be that there's something unique about the structure of the oligosaccharides in the mothers. It could be just a matter of seeding, which kinds of microbes are seeded through the environment very early in life. But in any case, this is now a strongly selected microbiome because of the HMOs, and it sets the stage for everything else that happens. And some of what happens now is most clearly observed in the gram-negative bacteria, which again, as I mentioned, there's a lot of Bacteroides in Finland and Estonia. These are gram-negative bacteria, and they're playing the role of utilizing the milk oligosaccharides. Whereas in Russia, we see much less gram-negatives in general, especially in the first year of life. And one thing to notice that the E. coli in red is actually fairly similar across all of these countries. But the ratio of E. coli, particularly to Bacteroides species, is very different. The ratio is much higher in Russia than it is in Finland and Estonia. And in terms of immune development, of course, gram-negative bacteria play a very important role. They produce lipopolysaccharide, or LPS, which is a very important immune stimulatory molecule, kind of classically an inflammatory molecule that drives TLR4 signaling on cells by the innate immune system. And so actually what's really interesting is in the old literature, it has been known that the structure of LPS from E. coli is different than that of Bacteroides, particularly in the lipid A moiety of LPS. The E. coli has 6 acyl chains, and the Bacteroides has 4 or 5. And so in collaboration with Novartis, we purified LPS from actually these strains that come from the infants, were isolated from the infants themselves, in order to understand their potential functional differences. And so what I'm showing here now is just the production of 2 inflammatory cytokines, TNF and IL-1, in response to large differences in concentrations of LPS. At the bottom, you can see the E. coli, which drive a very strong cytokine response, even at very small concentration. And then orders and orders of magnitude higher concentrations of LPS, you don't see any kind of a response from the Bacteroides species, particularly Bacteroides dorii, which was the most prevalent Bacteroides species in the Finns, and almost completely absent from the Russians. And here we're just performing a NF-kappa B reporter assay showing that this drives a kind of an NF-kappa B response. You can see at the very left curve is E. coli driving a very strong NF-kappa B response. And at the very bottom in that light purple is Bacteroides dorii driving a very weak NF-kappa B response. So the Bacteroides LPS actually does not seem to drive much of an inflammatory response at all. And so now we did some experiments where we combine different ratios of the Bacteroides versus E. coli LPS in vitro assays, where we put them onto these human PBMCs, and then ask how much inhibition of cytokine production do you see relative to just E. coli alone. And at a 1-to-1 ratio, you see about a 50% inhibition of these cytokines. But at a 10-to-1 or 100-to-1, which is more similar to the ratio of these that you see in the Finnish gut, actually you have almost complete inhibition of the production of these cytokines, suggesting that Bacteroides LPS is actually an agonist of TLR4. And if you have a lot of Bacteroides LPS present in the mix, that means actually E. coli LPS is non-functional. It's kind of rendered non-functional and cannot induce this inflammatory response in cells. And so actually, there's an important concept with innate responses like LPS, is that there is a endotoxin tolerance. So when you see it once, you see it the first time, you have a response, and then you see it a second time, and you have no response. The cell has kind of learned not to respond to LPS anymore, which is important because when you receive the exposure once, that's enough, and when you keep being exposed to it, your immune system is not going to go out of control. And so we did an experiment in these PBMCs where we exposed them to either E. coli or Bacteroides dorii LPS, and the following day we restimulate the cells with E. coli LPS. And this is what we observed. First, if you look at the hatched boxes, the E. coli, when we give the E. coli at day 0, the subsequent response, subsequent days, even at very high doses of LPS, you don't have any response in terms of TNF production. Now, if you only have Bacteroides exposure on the first day, then the following day you receive a very strong TNF response when you re-stimulate with E. coli, suggesting that Bacteroides is again preventing this normal kind of endotoxin tolerance. That means when you're exposed to another inflammatory, LPS later on, these cells are going to respond very strongly. And so we wanted to see in a mouse model of diabetes whether this can change the trajectory of disease. And we injected these NOD mice, a model of type 1 diabetes, with purified LPS from Bacteroides dorii or E. coli for several days, and then observed them. And the mice that received the E. coli LPS actually were protected against developing diabetes. And this might seem counterintuitive because E. coli is the inflammatory molecule, and so shouldn't it be driving this kind of strong autoimmune response? And so it's counterintuitive because it's actually E. coli is playing an important role in early life in allowing the immune system to learn and have this kind of endotoxin tolerance effect. Whereas if the microbiome is overloaded with this Bacteroides type LPS, the immune system doesn't have a chance to build that endotoxin tolerance. And later on in life, when it's exposed to a more inflammatory event, it has a higher chance of building into diabetes in people that are genetically predisposed. So this again appears to be directly related to diabetes in animal models. And so to end this part of the talk, what I'm proposing here is that the microbiome in early life between these different cohorts is quite different as a product of the many more exposures that the Russians have. Their microbiome is quite different. One of the biggest differences is the higher amount of bifidobacteria serving as human milk oligosaccharide metabolizers. And then later in life, this leads to this difference in the overall balance of LPSs. That means that the overall balance of LPSs is more driven by E. coli in the Russians and can lead to the induction of endotoxin tolerance, and the end result is a overall decrease in autoimmune activity. So this got me very interested in trying to understand what the microbiome looks like in different parts of the world, and perhaps more critically in different times, in different stages of human history. The Finland microbiome is kind of the ultimate industrial microbiome, the human industrial microbiome, modern human industrial microbiome. The Russian microbiome is kind of more on the non-industrial side, but there's nothing so special about it. There are many other communities in the world that are more relatively isolated from human industrial activity. But today, it's well accepted that there is nobody that's completely isolated from contact by industrialized communities one way or the other. The best evidence of this is the COVID-19 crisis that has stretched to every corner of the globe, even the most isolated communities in the Amazon, for example, where it's become quite a serious problem. So if viruses can spread, if COVID can spread, many other viruses can spread, and then many other microbes can spread as well. So in order to find a really, truly human microbiome that's not been tainted, so to say, by the industrial human microbiome, we have to go back in time. And so what did the pre-industrialized human microbiome look like in ancient human individuals? And just as a quick side note, I've been speaking about autoimmunity, but this is not the only kind of disease of relevance here. There are many other chronic diseases that we see increasing over time that we think is correlated to our industrial activity. And of course, another one is obesity and type 2 diabetes. We know that in the United States, obesity has been increasing dramatically for more than 100 years. And that the incidence of type 2 diabetes it has been increasing exponentially in the past several decades and continues to become worse. Quite a serious problem here, and I understand actually a very serious problem in Mexico as well. And I just show a plot here of where I'm showing that the incidence of type 2 diabetes is generally more prevalent in urban communities relative to rural communities. Again, there is something protective even in this metabolic disease with living in a more rural kind of a lifestyle. So to start to understand the ancient human microbiome, this was really work that was led by an amazing PhD student in my lab, Marcia, who has been responsible for coordinating the vast majority of this work. And what we studied actually was ancient human paleofeces or coprolites. So these are human stool samples that we had access to in very dry caves in North America. And we were kind of led to these samples by Stephen LeBlanc, who is at the Peabody Museum, now retired, who helped us actually getting into contact with so many of the collaborators that made this study possible. And so what he helped us identify was a set of samples actually twice as large as this list. This was the list that produced the high quality microbial DNA, but helped us identify many samples throughout North America. In the end, what our analysis is focused on is on 3 samples from Zape in Mexico that are about 1,000 years old. Another 3 samples from around Arizona that's also about 1,000 years old. And then 2 samples from Boomerang Cave in Utah that are about 2,000 years old. And all of the ancient DNA work here, the extraction of the DNA was done in the ancient DNA facilities at the University of Montana, led by Meredith Snow, a really great collaborator of ours. And so we worked with Christina Warner's lab in trying to authenticate these coprolites, demonstrating that they are really human and really of ancient origin. And so I don't want to go in too much details for how this was done, but essentially it involves verifying that the sequences come from actually the coprolites come from humans and not other animals like dogs that can leave samples in these caves, and that we can assemble them into microbial genomes that are consistent with what's seen in humans and not other animals. So we started with a total of 15 paleofeces. 11 had poor assemblies, just not good enough DNA, or 4 did, and so we were left with 11. We were left with 9 after removing soil contamination. 2 of them just had too much soil contamination to be useful. 1 was not likely to be human, and that left us with 8. And from these 8, we can see evidence first by carbon dating the kind of precise time period that they were from, so that we know that they're ancient. And also we can identify some small amount of human DNA in these paleofeces, and we can identify the haplogroups, which are consistent with the people known to inhabit this part of the world at this time. The authentication also involved electron microscopy and looking at food particles. We can notice things like metaspores, corn pollen grains, and cactus particles in these paleofeces, which is consistent with them having a human diet. And this collaboration was done with Carl Reinhardt. We also looked for the presence of other organisms in the stool, parasites, that were present in these to be sure that they're consistent with human parasites and not parasites of other animals. And what we saw, for example, was a lot of blastocystis, which is known to be associated with humans, as well as other parasites we found here that are human associated parasites. Another important part of the authentication was demonstrating not just from carbon dating, but from the actual DNA itself that it is ancient, and a telltale mark of being ancient is the damage to the ends of the DNA. So the ends of the fragments become damaged over time, especially with exposure to water, and they undergo C to D transitions, and that's G to A transitions on the other side. So we expect to see at the terminal ends, you can see the positions of the bases here. The terminal 5 to 15 bases should be highly damaged in this very kind of predictable pattern, which if you see it, it's ancient DNA and not modern DNA. And so we were able to demonstrate that. And also at the end of it, we got fairly large fragments of DNA. The largest that we have seen, and I think that has ever been published before in ancient human microbiome DNA. So whereas previous studies of the human microbiome in ancient people, you have to do kind of amplicon based sequencing of microbial genes. Here we can actually do shotgun sequencing and just sequence everything, which gives us the ability to reconstruct genomes. And so finally, we wanted to be sure that of the samples that we were working with, that they were not soil contamination. And here is how our ancient samples compare to known archaeological sediment. They're quite distinct. We show here soil samples that we took from Boomerang Cave, so we can be sure that that actually does look very different from the coprolites, and much more similar to known soil samples. So we felt pretty confident there. Of course, we also needed samples from modern humans that live a relatively, at least non-industrialized lifestyle. And this was one of the aspects of the study where our collaboration with Lorena's group was key. And this was actually very hard work, and led by Paco and Umberto and Cecilia and Angelica, where they went to the Nahua community in central Mexico that live a relatively traditional lifestyle and have a kind of very limited exposures, relatively speaking, to an industrialized life. And this was a very difficult effort, which involved coordination of keeping all of these samples frozen. If you can imagine going out to that community, having them, it kept frozen and shipped to Boston, by FedEx. It was quite logistically difficult work, and they handled the IRBs and a lot of work involved here. And so we're incredibly grateful. This was the only modern human stool samples that were included in our study. Other than that, we reanalyzed existing data sets of microbiomes from people in industrial and non-industrial communities. And so here's the really striking thing is when we look across all of these samples, all of the modern human samples, we see this very big difference, this kind of dichotomy of industrial, which is on the left here, and non-industrial, which is on the right. And the difference is glaring. And these non-industrial communities come from all different parts of the world, from South America, North America, Africa, Asia. So really remarkable how similar they are to each other relative to industrial populations that come from the United States and Denmark and Spain. And of course, what's really remarkable then is that the paleofeces, in fact, look much more similar to the modern non-industrial than the industrial communities. And the composition of the microbiome in the paleofeces looks like the non-industrial humans, but even more so. So it's known that the Firmicutes phylum is higher in non-industrial people, but then look at the ancient. They have even more Firmicutes than modern non-industrial. Same with the proteobacteria. Conversely, the Bacteroidetes, again, which in our Finland cohort was kind of, we think, a smoking gun, in one of the reasons why they had a lot of Bacteroides and why they have a high incidence of type 1 diabetes. It is much lower in modern non-industrial people, and you can see in the ancient really very low compared to modern people. So the microbiomes look strikingly different, and again, it's so interesting how similar the ancients look to the non-industrial, even though the ancients are kind of an extreme. And so you don't have to read the text here. I'm going to go through this in a little bit more detail, but here we're comparing the taxonomic composition at the species level across the paleo versus industrial samples, and then the industrial versus paleo feces samples. All of the taxonomy that were statistically significant. And you can see there are many, and the patterns are very clear, right? So zooming in on this, the thing that jumped out at us right away was Treponema succinificiens, which was present in every single one of our paleo feces, and very highly prevalent across the non-industrial world. It was absent in the Nahua for some reason. I'm not sure why. But if you look at the industrial, completely absent, was not present in even a single person from our hundreds of industrial samples. So this is something very clearly that was an important part of the ancient human microbiome that now is completely gone. And so we see a similar trend with Prevotella, and we see a similar trend with these other four microbes that are Firmicutes. They're all related because they are short chain fatty acid producers that produce short chain fatty acids from fiber, dietary fiber. So they depend on a high fiber diet, and they're able to convert this fiber that's not digestible by us into these very beneficial molecules, short chain fatty acids, that play important roles in many aspects of our physiology, immunity, metabolism, and many other roles. So I highlight those, especially with the Prevotella, which we know tend to be much rarer in industrial life. The Prevotella are again present in every single one of the ancient human microbiomes. And then conversely, we see things like here, this Egrithella and this example of the Bacteroides, almost completely absent in ancient, but very highly prevalent in the industrial world. Akkermansia mucinophila, this is actually a mucin degrader that degrades the mucus layer of our gut. It is very prevalent in industrial countries, much less prevalent in these non-industrial communities. And Bifidobacterium adolescentis, for some reason, actually is completely absent in paleofeces. We couldn't find this bifidobacteria. So perhaps their human milk oligosaccharides utilizers were different. As I mentioned, because we had such large fragments, 300 base pair long fragments of these ancient genomes, we could reconstruct them. And this was done with the help of Nicolas Segata, who's an expert in human metagenomics. And we were able to get 181 good quality genomes. And what was really surprising is that 40% of them were completely new species, never seen before, new species of bacteria, suggesting that the ancient human microbiome had many species that no longer exist, and suggests that perhaps these microbes may have gone extinct over time. And we can see, with the help of Frank here, the phylogenetic distribution of these ancient microbes. They are, of course, all over the map, different phylogenies. They belong to many different classes of bacteria. We wanted to understand the evolutionary history some of these ancient microbes relative to modern microbes. And to do this very actually complicated work, we enlisted the help of Omar, who is in Italy, and the help of his really fantastic graduate student, Kuhn, who built us these trees. And we focused on Methanobrevibacter smithii because we had two really high quality genomes from two different samples, and compared them to Methanobrevibacter smithii samples from all around the world, modern samples, of course, isolated from humans. And what we see here is, if you see the branch here in orange, it's separated by about 27,000 years, our ancient North American Methanobrevibacter from all of the modern Methanobrevibacter. And so what's interesting is that that time scale coincides very well with human migration into North America that happened, you know, in this time frame between 20 to 40,000 years ago, when humans crossed the Bering Strait from Asia into North America and then into South America. And so this is evidence that Methanobrevibacter smithii was vertically transmitted across all of these populations over, you know, thousands of years. So we can almost think of these Methanobrevibacter smithii genomes as part of the human genome, because this is kind of natural selection at work. These are genes that go along with us from mother to child for thousands and thousands of years. So think about natural selection in the human genome, it actually might be happening on our microbial genomes as well. So we think that this is, to us, a really exciting result from this ancient human work. And of course, having the genomes, we can also look into functions that are different between these microbiomes. What we saw enriched in the paleofeces and in non-industrial people was a lot of transposases. A lot of transposases suggesting a lot of horizontal gene transfer events in the microbiome in those communities that appear to be very rare in industrialized communities. We also see, as expected, antibiotic-resistant genes very prevalent in industrial communities, not so prevalent in non-industrial communities, and completely absent from the paleofeces, as well as things like glycan degradation, which relates to mucus, the mucus layer degradation, Fimbriae on bacteria, which are allow bacteria to attach to human cells. They kind of serve a pathogenic function, completely absent in the paleofeces, and more antibiotic-resistant genes, which of course makes sense. And then finally, looking at the carbohydrate active enzymes and looking at the roles of these enzymes and kind of focusing in on the middle plot here, all of the industrial enrichments on the left and the paleofeces enrichments on the right, we see a lot of starch and glycogen functions in the ancient microbiome. So actually, ancient individuals did eat, and this is confirming other recent studies, quite a lot of carbohydrates, not just animals and fish, of course. They also had a lot of chitin, which is consistent with a diet high in mushrooms and also in insects. And we do know that insects played a large role in the diet of ancient peoples in North America. On the other side, in the industrial side, we see a lot of mucin degradation. Again, degrading the mucus layer of our intestines, which is a pathogenic function. Interestingly, we also see alginate, which is seen in seaweed. Maybe this is because eating sushi has become so common in the Western world. I don't know, but that was an interesting thing to see as well. So the functional repertoire of genes in the microbiome of ancient individuals is quite different. So future goals, we want to understand whether these microbes have really gone extinct, and whether when we look at different time periods of human history, different parts of the world, hopefully in the future we can look at South America, and we can look at Africa, in addition to many more parts of North America, we can start to have a lot more confidence in kind of this idea of a shared universal ancient human microbiome, and more evidence that these microbes are going extinct in modern life. We, of course, are very interested in synthetic biology applications and trying to identify genomic regions that are only present in ancient human microbiomes, and trying to resurrect them into modern human-associated microbes. And then, of course, we are very interested in how these can be translated into therapies, particularly in type 2 diabetes and obesity, which is kind of one of the most important applications in our lab. So with that, I'd like to close, and I'd like to thank the many people that were involved in this collaborative work, who I mentioned throughout, and I'd like to thank them, as well as my funding sources that I've listed here. Thank you so much for your kind attention, and I look forward to receiving your questions. Thank you very much.